When it comes to the 3D printing, having some advancements in the 3D printer space, particularly for the pharma manufacturing, is very, very important.
Welcome to Data and Biotech, a podcast from Cornell where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
Jaidev Chakka, welcome to the Data and Biotech podcast.
Glad to be here.
Awesome. Well, just to get us started, would you mind giving us an introduction to your background, what brings you here to the AAPS PharmSci 360 conference?
Yes, I'm a principal scientist at University of Mississippi, and I'm working with Dr. Mohammad Rajuman in pharmaceutical engineering and 3D printing lab. So this is my third time coming to this conference, which is a great venue to meet all the pharmaceutical scientists and researchers from academia, industry. Everybody come together and share their experiences. So it's a great event here.
Yeah, awesome. And would you, we're going to spend a lot of time. So you mentioned 3D printing. Would love to hear just a little bit about like your research interests and some of the things that you've done in your work today.
Sure. Yeah. 3D printing is a unique technique where for me it is like printing something or creating something out of nothing. So you have a filament, which is a non-living matter. And when you put it in a 3D printing, it comes out of into a beautiful shape. And you can define the parameters of the shape, you can design it, you can control it. So that's what it fascinated me initially. And then when I got my hands on the 3D printer, then I thought like I have a background of biotechnology. So how can I apply that for biomedical applications? So that's when I started making scaffolds for bone regeneration, tissue engineering, 3D printing back in India when I was a postdoc at the Indian Institute of Sciences in Dr. Kaushik Chatterjee's lab. After that, I got an opportunity to work at University of Iowa under Dr. Aliasgar Saleb. So there I worked on gene therapy strategies. So now, taking a step ahead from a scaffold that just gives shape and structure, we're able to give a treatment in terms of functionality to that. So that's a great addition in terms of progression that I see how the 3D printing as a technique is evolving, right? And now in the present lab, I'm actually working on not only just making 3D structures and not only giving it functionality, but we are able to have cells, particularly spheroids and organoids, that there will be, the scaffold is acting like a host, giving space for the cells to grow inside them. And this whole architecture, even though it's super complex, it acts like a kind of micro tissues, which can be transplanted for regeneration purposes and can be used for preclinical testing, disease modeling, lots of opportunities.
Interesting. Yeah. So I know that the applications of 3D printing to biotech are sort of growing and in the process of coming into be. I would love to spend some time on some of the use cases that you just mentioned to understand them a little bit more deeply, and then maybe we can talk a little bit about sort of the how data and machine learning and AI come into these applications of 3D printing. Is that all right?
Yeah.
So scaffolding for bone generation. Would you mind just introducing us to the situation where like a 3D printed scaffold for bone generation would be useful and like how it compares to the way that we would typically do that sort of thing?
Sure. So the gold standard for a bone defect or for the accident case or something is an autologous graft. So a part of the femur bone taken from and then put it in the ribs or some other place where it got broken can't be cured naturally, innately. So such defects generally called as critical defects. So anything bigger than like 8 mm in terms of the defect volume, diameter or length, depending on the region, those are critical defects and they won't reunite back. The broken two joints, they won't grow and reunite back by themselves. So you need to give some support. So that's where autologous grafts come. But again, it's not like you're taking some part of the body and putting it somewhere else, but still there is a loss of the component of bone in terms of mass overall. Now with the MRI scans and CT scans, you can actually regenerate the void that is there in the defect region, and you can print exact graft that fits perfectly there. And now having the material aspect of this 3D printed structure give you the strength and shape. So the body, because it's biocompatible, the body will immediately accept it. And then the cells that they migrate onto the scaffold because they can identify it as something that they can rely on and they can grow. So that's how the cells migrate and the whole regeneration process happens.
Yeah, so let me say that back to you to make sure that I'm understanding. So, typically you take bone from, another being somewhere else and you put it in the body, and then, but anything longer than like 8 millimeters, it's the growth isn't going to happen. So you're 3D printing this scaffolds that can be longer, it can be much larger, but it's biocompatible. People's bodies will accept it. But the beauty of the scaffold is in the same way that we scaffold a building, you're scaffolding the bone so that the cells can sort of populate the gaps in the scaffold and like grow into the fully fledged bone. And then the scaffold, I guess, is consumed or falls away. Is that sort of what happens? Or does it stay there as a scaffold or
So
pet?
yeah, just one correction.
Sure.
In an autologous graft, the bone won't be taken from the other patient. It will be taken from the same patient to avoid immune rejection.
Yes.
So when it comes to the scaffolding, there is no chance of immune rejection because there is no component of that is present. It's just inactive material.
Yes.
Right. And coming to your question, that's a good question because the scaffold, if you take titanium kind of materials, making it into a 3D structure and putting it there, it stays there for eternity until the patient die. It stays there. If we take the biocompatible materials like polylactic acid or some other materials that has these thermoplastic materials like PLGA and all, so PCl, PLGA, so they actually degrade slowly in the body, not at a faster rate, but very slow. It's like 1 mg of these materials take 2 years to completely decompose in the body. So that way, slowly they just keep on replacing, the cells replace this material with the actual calcium deposits that they grow over the time.
Yeah, that's amazing. So you're literally just giving the body time to rebuild the bone, to rebuild that bone itself and the strength. Moving on to, gene therapy strategies. Can you help me understand sort of the intersection of 3D printing and gene therapy and sort of like how that works?
Sure. As I mentioned in the example, like the cells, they need to migrate. So cells, obviously, they want some stimulus, like we are giving chocolate to the kids to listen to us, something similar to it. So when there is nothing like a void space, it's not exhibiting any characteristic bone properties. So the cells sometimes may take longer than usual to migrate onto these defects, even though you do a 3D printed support. Now, to accelerate that process, for the cells to grow, come together and grow, we give some stimulating factors like bone morphogenic protein, also PVDF, VEGF, and fibroblast growth factor. So these things. So when we put these plasmids, even though 1 or 2 cells come, they just express these plasmids, giving the protein out from the cells. Now, these act like a chemokines where the cells attract towards these chemokines and come back. So from 1 cell to like 1,000 cells to like million cells. So that way it accelerates the process.
And I think what I'm missing is sort of like the intersection of that with the 3D printing technology. So what are you 3D printing in that?
I answer your question. Yeah. So 3D printing, fused deposition modeling 3D printing that we do, it involves high temperatures, 150 to 200 degrees centigrade. No biological component will be alive after coming into that environment. Right. So we'll separate the things. First, you print the scaffold, and now you coat the scaffold with the biological gene therapy materials.
Yeah.
So again, how good is that coating? So for that, I used polydopamine. Polydopamine is a proteinaceous matrix that is exhibited by mussels. So mussels are generally considered as a hull of the ocean because they stick to the ships, they keep degrading it, they reduce the flow of the ships. So cleaning them is like a big hazard. So if you analyze that mussels, that sticky nature that they have, what is the actual component that is allowing them to stick to the ship even though it's speeding at like 100 knots or what? I'm like, I don't know. I'm just assuming that is the polydopamine component. So luckily this dopamine component we can polymerize from the individual dopamine hydrochloride units by manipulating the pH. So now we take the scaffold, put it in a dopamine hydrochloride solution and then change the pH. When you change the pH, automatically the dopamine polymerizes. Because it comes into the contact of the scaffold, it has its ability to stick to any kind of material. It sticks to the scaffold. And because of this nature, the gene therapy plasmid complexes with the PEI also sticks to the surface. So now this dopamine coating is actually like a mediator that is like a double-sided tape for both the scaffold and for the plasmids.
Okay, very interesting. So you're sort of enhancing the scaffold to make it make it more functional inside of the body by coating it in this quote unquote double-sided tape. Okay, that's very interesting. So my understanding, there are at least a few companies here that are doing 3D printing for like manufacturing applications. Can you explain from your experience, what are some of the most common or the most valuable like applications of 3D printing to pharmaceutical manufacturing?
So for pharmaceutical manufacturing, the lab and the PI, Dr. Mohammad Rujwan, he's actually initiated the 3D printing process for pharmacy applications. So he envisioned a thing like in a home, there will be a printer that continuously printed the formulations those are tailored to the patient. So that's the vision that was given to us. So coming to this pharmaceutical manufacturing, 3D printing is a unique tool where you can put multiple drugs with multiple materials into a single tablet. So in a traditional manufacturing, you have to evaluate every component. For just the drug to go into the body, you need to give an excipient that will shield the drug and protect the drug.
Right.
And during the manufacturing, you need to give some other components because when it compressed, it has to come out. So you need to give some material. And for the compressed, in order to maintain strength, you need to put another material. And for the flavoring, you need to put another material. If you want to give a color, you need to put another material. All these things goes on. But whereas in 3D printing, it is just the material, the material that shield the drug by compatible like HPMC and all, and the drug molecules. That's all. And the heat that the entire process undergoes, it simulates in a way, not exactly, but the hot melt extrusion process where it amorphizes the drug. So this particular kind of technology is mainly suited for personalized medicines. For example, a patient, the commercial tablets, those are like fixed doses, like 100 mg, 500 mg. But for some patients' condition, they don't need 500 mg, but they want more than 100 mg. So the optional, let's say it is 200 mg or 250 mg, that is not available. So there has to be like, okay, break the tablet into half and then take only half tablet. Right. But here we can actually tailor that. Like, okay, this filament of this material of this concentration, if you give these dimensions of the tablet, you will get this dose. Like that, we can actually tailor the doses for the, and for the manufacturing side, it is challenging in a sense of speed and scale. Why? Because the tablets, they're currently manufacturing like thousands of tablets per minute, whereas 3D printing, one tablet will take like 3 to 4 minutes for printing sake. And if you want to scale that, and like say one printer print one tablet at 4 minutes, if you want like 1,000 tablets, in order to get optimization wise, you need to have 1,000 printers. So all the 1,000 printers printing at a time. So you'll have 1,000 tablets in 4 minutes. Now, if you want 10,000 tablets, right? So that scaling aspect is what I feel like currently it is limiting in terms of like even trying it out at a mass scale. But the technology is evolving in terms of 3D printing, and also pharma companies are also exploring other opportunities. So one aspect that I observed is they're going into continuous manufacturing. So with the continuous manufacturing, there is no batch. So it's like materials are continuously coming into the reactor and they're making it and then just getting it out. And that way, I don't know, but the report or what I read, it shows it saves a lot of cost for them in terms of operations wise, right, for the company. So if we adopt that, we did one study where we compare batch printing versus continuous printing, which one is best? In that study, we used AI to see what parameters are actually helping. What we see was continuous printing is better than the batch printing. So we are evaluating further like why is that? But the two papers that we got from the lab, we published from the lab, both show that continuous manufacturing is a step ahead from the batch kind of manufacturing. But it is with the 3D printers, of course. But yeah.
Right. So you mean using 3D printers to print in batch versus using 3D printers to print like continuously, like one by one?
One by one.
Yes. Okay. So that sort of makes sense. So I don't know. Some of the things off the top of my head that might make continuous manufacturing better than batch are, when you manufacture in batch, you have to sort of statistically sample from the batch in order to determine whether the batch is working or not. And then regulations force you to throw away the batch if you exceed a certain failure rate. Whereas with continuous manufacturing, you can sort of understand on a one by one basis what are the predictors of failure and maybe like throw away one dose, or you can detect that there's failure that's happening as you're manufacturing it, and maybe even alter the process parameters to avoid that failure, like in the middle of manufacturing. Am I thinking about that right? Or what are some of the other like contrasts that you would draw between like batch manufacturing versus continuous manufacturing in that?
Yeah, you're absolutely right. So until now with the batch, like if one batch is gone bad, so you can throw it. But with the continuous manufacturing, when you do the direct comparison, it seems like, okay, that may be a problem with the continuous manufacturing. But in the continuous manufacturing, when the process is going continuously, there are no other factors that are coming in. For example, in batch, once the batch is done, you have to clean it. You have to get it ready. And in some error somewhere happen in terms of the cleaning is not good or something. So which means the next batch is going bad. So, but in continuous, once you set everything, it's going continuously, right? There is no breaks in between for them to any contamination or some variation or something. So as long as the material is coming up in a consistent manner, so they were able to do that reaction and get the material out in a proper manner. So once it is regulated and it is deemed right quality, I think, it goes continue. It looks like a good batch.
Yeah, that's it. That's interesting. It's not necessarily intuitive to me why batch machinery would be more likely to fail or less reliable than continuous process manufacturing machinery. Is it that in continuous manufacturing, there's like specialization in the components that are happening along the lines? Like, because I would imagine that like any individual like process step could also fail, but maybe I'm thinking about it wrong because when you're comparing 3D printer to 3D printer, am I making sense in terms of like
I understand your point, but I think when it comes to the 3D printing and from the traditional manufacturing, obviously the processes, everything is different. Drawing a direct comparisons may not be an ideal case, particularly in this scenario.
Yeah.
But I think when it comes to the 3D printing, having some advancements in the 3D printer space, particularly for the pharma manufacturing, is very, very important. For example, FabRx, they're doing great in terms of making a GMP grade printer so that can print doses. Right. We are also in line to do something like what FabRx did. So where we can have our own GMP printer manufactured and designed in USA.
I want to return sort of the 3D printing and the data that you collect from 3D printing so that we can understand sort of like the applications of ML and AI. Can you give us an insight into like what are some of the data features that come off of a 3D printer that help you to understand what's happening as you're manufacturing? Sure. Yeah, pharmaceutical.
We work with the fused deposition modeling method, and there are different configuration of the printers that work with that particular principle, like Core X and Core XY, different configurations right now. So the Cartesian printers and all. So the configuration may be different. Delta printers is different, but the principle is same, where you take a hot plastic filament and you melt it and lay into it a shape or structure. That's the core working principle of the 3D printing.
And it's like layer by layer. Yeah.
So in this process, when you observe like the printer speed is one parameter, and the print temperature is one parameter, and the flow percentage is one parameter, and the material that you are printing is another parameter that we took. When we printed, we normalized the material because it is in direct correlation with the print temperature. Now the print speed, print temperature, infill percentage also took infill percentage, and flow percentage. So what we did was we created combinations using DOE. Is enough experiments what we did, and we find like what are the least combinations that we can get to get an understanding of zero defects or something. We got like 81 combinations. So that's a huge data set that we need to make, because we need to print, we need to convert that printed tablets into readable data for the algorithm. So we used a non-drug loaded placebo filament. And the placebo filament, what we used, and we made these 81 combinations, and then we printed. And a graduate student named Jaidev Chakka, he's instrumental in spearheading this work. So when we did that 81 combinations, we took images and we took all the parameters of the tablets, and when we put this into the adaptive design, so these 4 parameters, what I mentioned for the printer, what value of each parameter can give us 0 defects. Like the tablet should be solid, no defects, no pores, no holes, nothing. Right. That's the end goal, because that's the quality parameter we want for the final tablet, right? So when we put all the data into the algorithm, it came out. Flow percentage is the key value, not print temperature, not print speed, not infill percentage. Is the key value to get 0 defects. So in the hindsight, we know all these go hand in hand, like these 4 parameters go hand in hand in order to give a good quality of the outcome from the 3D printer. But it is surprising to see the flow percentage is what the value that is important for getting a good print, because print temperature goes with the material, print speed goes with the printer. And in this case, print speed has not major role, to be frank. In our mind, we thought like when it is printing at high speed, there may be a chance for more defects. At low speed, it may be good. But here, based on the predictions that we got from the algorithm, an inflow percentage with a little bit higher value at higher print speed is giving us better quality. In a way, that saves time, and we can get more tablets printed in less time without compromising the quality of the printed tablet there.
Can you maybe unpack what flow percentage is and what the intuition is for why that would be like the most important determinant of the quality?
So the flow percentage is generally considered for water. In the 3D printing space, the molten material has flow properties, but not the hard filament, because it can't flow, right?
Okay.
But the molten material has flow properties. And now how much material the nozzle is pushing or flowing out through it is what that percentage value. That percentage is again calculated from the filament diameter and the nozzle diameter. And then from these two values, they make a lot of mathematical calculations, but that calculate that percentage.
Right.
So
like the percentage of how much it could potentially flow out, or
yes. So the filament is constant, the diameter is constant, material is constant. There is no variable to there. But how much the material is coming pushed towards the heated nozzle. So if you push more, I mean the extruder, if it is pushing more, you get more material out of the nozzle. If it is pushing less, then it is less. So considering all these different components, so the software gives the final like the percentage value. So if you alter the percentage value, it says the extruded to push less, and obviously the material comes out of the nozzle also less. Yeah. So we played with that material to see that value. For example, in the manufacturing scales, let's say if there are some defects are happening, then obviously you can guide it like, okay, push a little bit more in order to fill the space, so there won't be any defect.
That makes a lot of sense. So, so if I'm thinking about that right, then hypothetically you could have the 3D printer like adapted in this case to the flow percentage to manage the flow percentage to stay relatively constant, and that like the implication there would be that you would minimize defects by keeping the flow rate constant.
Yeah. Okay.
Are there any other sort of like machine learning and AI applications to 3D printing that you think are really relevant or exciting?
Two things that I feel like exciting. We used AI for understanding what parameters are relevant to get a defect-free printing on a 3D printer, right? So now, can we apply the same design in order to understand what are the key parameters that influence the release of the drug? It's not like we don't know, but when we have a data set with hundreds of papers, not only hundreds, to be frank, in the pharmaceutical research with 3D printing in the last decade, you find like so many papers, right? Yeah. There was a group in UK, Dr. Bassett. So their group actually published a paper where they used the literature as a data set, and then they did AI algorithm, and then they predicted certain combinations in order to see what the outcome in terms of the release profile. That was done, but taking a step ahead, can we combine in terms of manufacturing and also in terms of release profile? So we are in the second leg now. So to see if we give for these predicted parameters on the printer, if we give a different materials and the drugs, so how the what are the predictions that the algorithm can give in order to get a defect-free tablet that gives a tailored dose and a release profile that is suited for the patient. That is one interesting application that we want to move ahead into, right? And the other one is, on the hindsight, we are also working on spheroids, cell spheroids, mesenchymal stem cells. For the bone regeneration I mentioned, I use mesenchymal stem cells for differentiation towards osteoblasts. The stem cells, as spheroids, are they different or are they same? So one understanding, even though not published, but one understanding what we have is, after making HMSC spheroids, they're way different than the regular HMSs in terms of retaining their stem cell nature. Cells, particularly primary cells like mesenchymal stem cells, the passages, like they grow and then you split them into multiple flasks for them to grow more. So that each passage, they can grow only like 4 to 5 passages. After that, they quit. They differentiated into any other cell that they like, mostly to the osteoblast. But when you preserve them, when you make spheroids of HMSAs and you culture them like a week, and then when I grow them back in the petri dishes, the passage numbers were higher. So that's something interesting to me. Like, maybe in the body they are not a monolayer. Maybe when you put them in the native space, any kind of thing, maybe they are a bit more active and able to give more passages there. I mean, able to grow more generations. And there we want to have AI in terms of optimizing that process, because optimization is the key step in order to get a quality outcome, right? So we can actually work into different, again, parameters, the growth time, the growth media, growth conditions, morphology of the cell through fluorescence microscope imaging or confocal. And we can actually get an understanding on how these parameters are actually influencing the shape. If it is a spheroid, then it's like sphere growth, the volume, then a number of cells that we're getting there, all those things. And extrapolating the same to the organoid. So that's a very interesting space, because organoids are like tiny organs that give both that have both the cell phenotype of the organ and also the functionality of the organ.
Yeah.
So in a way, you're like working with the organs on at a micron scale in the petri dishes. Not exactly micron scale, but yeah, the petri dishes. If we can harvest the intelligence, if it is a neuronal organoid, they can respond to the stimuli, right? So when it's an electrical signal, there is always intelligence there. That was proved by computers, right? So these units, functional units, can act as an individual processing entities. So where we can actually create an intelligent systems around that. And to optimize those systems, like its behavior and also how much input, how much output, and what is that output, how to interpret that output. So I feel like the AML can act like a GPU for the CPU of these organoids on top of this application.
So first of all, I just want to zoom out and make sure that everybody's sort of tracking. So like the organoids are like, you mentioned, miniature versions of the organs on of an organ on micron scale, and then you mentioned that so one version of that is you can have an organoids like neurons that are firing, but have an electrical signal, and that electrical signal can be understood as computation, and so one of the applications of AI and ML to that computation is this idea that, you can use a machine learning model to train the organoids to produce like computational spikes. But I think what might help is like, what are some of the applications that you would see of like having organoids that could do that kind of computation?
One particular example from Dr. Thomas Horton group, it was like evaluating the toxicity of the developmental neurons. So, I think from my understanding, if I read the paper right and I understood right, generally for those kind of studies, you need to go with animal models. You need to understand how during the development, how these brains, neurons, everything work. If you put the toxicity, what happens? But when they have these organoids and with the AI adaptation where they used the electrodes that can capture the signal, and these electrodes can also transfer the signal back to the organoids. So these innovations are important, right? For particularly like facilitating these kind of studies, they were able to identify how these toxins are affecting the ability of these neurons to firing up and firing down. It avoided the use of animals per se for the animal care and also, yeah.
Yeah, no, that's really interesting, and also it hypothetically like a methodology for a new form of assay. Like if you can train the neuronal cells to like fire in certain situations and not fire in other situations, and you can, using AI and ML, control the parameters on which that firing happens, then you might be able to, similar to the way that like fluorescence is used to detect all sorts of things in biology. You could also sort of use this method to detect other things besides toxicity inside of these organoids. Am I thinking about that?
Yeah, your understanding is right. I mean, in our whole body, everything everywhere is there. What I mean is, if you take heart, cardiomyocytes are the major component, but you have blood vessels, you have neurons, you have other components like the epithelial cells and everything. So even though when we say we are working on heart research, if you take just cardiomyocytes, okay, that's good, but it's not complete. But with the organoids, we can actually either differentiate into other cells also, or with the iPSCs, in these pluripotent stem cells, we can actually differentiate into cardiomyocytes and try it out, because it preserves that innate nature of the cardiomyocytes rather than culturing them from few cells from the heart, right? So that's how those are more relevant. But increasing the complexity wise, now what we understood was from the organoids, you can actually measure the toxicity based on these hiring and firing patterns. Can we extend this to the other organs? So now having a brain organoid and a heart organoid and a liver organoid, and now when you give a dose to the liver organoid, what happens to these ones?
Yeah.
Right. So these kind of interesting thoughts at least give some kind of energy to enthusiasm to look into those directions. But it is possible with the kind of advancements we are seeing in microfluidic ways, and also the electrodes that I mentioned. Without those electrodes, we can't do that kind of studies, right? But there is a company that was actually doing that, Axiom System. So as time goes by, these technologies are going more mature and also going much advanced. So that gives at least a little bit of hope for somebody like me, at least to think in that direction, to see maybe one day I can do that.
Yeah, no, that makes sense. So you've got this multi-electrode array. You've got a bunch of electrodes that can both receive signal and can send electrical signals, and then you can connect that to a computer with a model on it, and the goal in a certain case would be to train this organoid to detect something that's happening biologically inside of the organoid system that you just described. Can you help me understand how would that training process work to the extent that it's known? I recognize that this is sort of like at the frontier of the field, but understanding how do you teach the neuronal organoid to send and receive the signals when it's detecting, for example, toxicity?
Yeah, it's already proved with various publications and by researchers. For example, one publication from Nature, I believe, is from Dr. Kagan, where they used brain organoids to play ping pong. So they were able to train that to making them to play the video game. Right. So it may sound silly if you hear first time, but when you see the research, I mean, it's fascinating. Yeah, the kind of things that they were actually doing, and even for the recent cortical labs, they were actually making a digital brain. So for like a preclinical evaluation kind of thing, where we can put different materials and see how the brain reacts. So instead of going with the actual animal models of the brain of a living being, right? So training these organoids electrical signals. So how much signal you are giving to that cell and how much you are coming out, how much the cell is giving you out, right? So when it comes to this particular memory aspect of these neuronal cells, they have kind of, on top of my head, I don't have all the information in terms of like the, for example, if you take the brain, the cerebrum, cerebellum, medulla oblongata is like a seating center for the entire brain where it stores the information and it processes it, right? So if we consider the same for the organoid, it won't be at that scale, but it is able to still process it, right? And when we give the training for the cell in order to store that signal for a fraction of time, that's their memory. When you give that command in a digitized form at a particular voltage to that array, to that organoid in the whole array, so that store only one bit of information. Now you have like 100 organoids storing 100 bits of information. And when you put that, I don't know how it is, that machine learning code in that electrical signal format, now these 100 odd neurons, some fire, some not. And the outcome that comes from there is what I think like an output signal for it to operate.
Yeah, I think I might need to just read up more on the subject. But the idea of sort of like gradient descent or of back propagation in the context of organoid learning is something that's both very fascinating to me, but also hard for me to understand based on where I am in terms of my biological understanding. But yeah,
no, you're good. I'm also learning so much. Yeah.
You've described your lab as sort of like in our previous call as taking baby steps into AI. For your lab and for the scientists that are doing work in your lab, what's like been the steepest learning curve in terms of like implementing AI and ML for your team?
Having the right people and the right people that they're able to understand what we are trying to achieve. For example, the paper that we worked on, the pharmaceutical dosage forms with AI, we worked with Dr. Samrat from School of Engineering at the University of Mississippi. And before that, we had like some data scientists, a couple of them, but their understanding is from the sake of data, from side of data, but not exactly like what we are trying to get. I mean, like if we give a bunch of information to them, they are ready to process it, but they're not able to ask us what is the data that I want in order to do this application for the pharmacy side. This professor has this understanding and he was able to ask us all the right questions and we were able to give him all the data that he needed. So that's how we were able to get that done. So the steepest curve is being a traditional biotechnologist or pharmacist or some, these are all like unexplored and new venues for us even to think in these directions. So yeah, I mean, that may be the steepest learning curve for us to understand who wants what and how to communicate to them to get the outcome.
Yeah. So, I mean, you have a particular like experiments you're trying to run or biological phenomenon that you're trying to measure, and this person's coming and saying, well, you need the person who can ask the right questions about what can be measured, about how it's being measured, about like what's being predicted and how to connect the dots between those things. Am I thinking that right?
You're right. Yeah.
As we come toward the end of our conversation, I'm just interested in, if we fast forward, 2 to 5 years, what breakthroughs do you hope to see in, AI-enabled pharmaceutical manufacturing or organoid development or personalized medicine that you think have the, the most transformational potential based on the research that you're doing?
We may see something in a direction called maybe like instant manufacturing, like instant coffee. Everything is ready. You just have to put all the ingredients there. Your coffee is ready. So something like that. So the filaments that I mentioned previously, when we load drug into them, they sort of protect the drug, keep them stable. So now when you have these filaments, like a cassettes, at your disposal in a particular mission that work with, like, for example, if you need aspirin, you're in the middle of the night, simple drug, but still you don't have it, you need to get it. So having a cassette of that, you can just press the button and there you have it. Or for example, some critical doses that you need to take in a defined, very, very complex doses. You can print them again back in the same printer, having these all filaments there ready at you. So we are actually devising in a different direction of same in a different direction, like having a printer that can print multiple doses at a time. Like doses in the sense, not like individual tablets of different doses, but also the same tablet with different doses of the drug, or also different drugs themselves. So that we call portable. There we need some software advancements, of course, with the integration of AI. That's going to be a beautiful thing to see. Because AI continues with the feedback mechanism as it is learning continuously, and also it can adapt to the changes that it's happening in a patient's health or so. So it can actually manage better, in my opinion.
I would just want to unpack that a little bit to make sure that I'm understanding correctly. So, we touched on personalized medicine earlier. I want to really open it up now. So when a person takes a manufactured dose of a pharmaceutical, let's say they're consuming it orally, it's decomposing, it's dissolving in their stomach, it's getting into their bloodstream. People are metabolizing it differently based on, where they are in their lives, based on what's going on with them health-wise. My understanding is there's this opportunity to sort of personalize in a variety of dimensions. There's, personalizing for you and your biology, like this is the dose that's meant for you, but then also, the dose that you should be taking, or the nature of the pharmaceutical that you should be taking, might vary from day to day and week to week. So there's this opportunity to integrate the porta-pill or the personalized manufacturing with sort of the status of the patient at any given point in time. Am I thinking about that right? Or what are the other sort of vectors of personalization that you would think about in terms of the?
I know you're absolutely right in this scenario. Like as the patient needs changes, the dose changes, and as the doctor prescribed them today take only 5 mg less than what it is what it was yesterday. I think that's possible with a portable. Yeah. And it's an integrated system where the vitals are continuously monitored by the physician. And any drop in them or any change in them, the physician will have the information of that, and physician can actually assign, he can instantly say, okay, take this mg of dose of this, and he can send it directly to the printer. So the printer will print and the patient is ready to take the drug.
Yeah. And then also, from a commercial perspective, you don't have, the pharmacy doesn't need to be open. It can just be a vending machine with a 3D printer in it. You just go in and this is who I am. I need my dose or something.
Yeah. It sounds. Isn't that futuristic, right? Yeah.
Yeah. I mean,
in one day maybe.
It feels right. And also, but there's so much work to be done on a variety of dimensions. There's the assays of, being able to understand what's happening with the patient without having to take, huge vials of blood and or use, very expensive mass spec equipment to understand what's happening. And then there's inputting that into the feedback loop of the manufacturing systems. And so, yeah,
you're absolutely right. Like the traditional manufacturing is thought of a lot of things, and that's it was actually it has standard process, and the USP, FDA giving the guidance documents, and also USP giving us the monographs on different drugs and other things. So over the years, so all these regulating agencies and also these particular bodies, they were doing fantastic job in order to have a normalization in terms of the use and also the application, evaluation, quality, everything. Now with the 3D printing, if I come and say, hey, you don't need all that, you just need doctor will tell you take that's not how it is. So we have to get these processes and also the evaluations incompatible with the regulating agency guidances, all those things. And then only we can move ahead, because ultimately everybody wants the patient to be having a good quality of life. But some may think like, okay, I don't need this. I'll do this way. No, that's not the case, because we have to ensure the safety and efficacy of the drug and the safety of the patient in every step that we are doing. So even though we are dreaming big, or we may be like having some bold ideas here, but still we have to do each and every step that is required to ensure the patient safety and also the compliance in terms of drug handling, excipient handling, manufacturing standards, everything.
Yeah. That makes a lot of sense. Well, Jaidev, I really appreciate you coming on the podcast today. Where can listeners go to learn more about your work?
They can go to Google Scholar, so LR Jaidev. If they type that, they can get all my publications there, and also LinkedIn. The same. Yeah.
Awesome. Well, Jaidev, thank you so much for coming on the podcast today. I really appreciate it.
Thank you very much for having me. I appreciate it. Thank you.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.